Good morning all. This is the 5th day class in respiration. In previous class we discussed about structure of mitochondria, transportation of oxygen from alveoles to blood, types of respiration. And before going to start today's lesson, you have to answer these questions. Draw a diagram of mitochondria and explain about its structure. Write differences between aerobic and anaerobic respiration. What is the reason for getting muscle pains after doing severe exercises? And today we want to discuss about one important topic. It is about anaerobic respiration. This anaerobic respiration also called as fermentation. Fermentation is a process of production of alcohol with using of microorganism in anaerobic respiration. In anaerobic respiration, microorganisms are converting glucose into alcohol. This process is called as fermentation. We want to prove carbon dioxide heat is released in anaerobic respiration. To prove this, we want to do one experiment. To prove that anaerobic respiration or fermentation in yeast cells. Aim. In this activity, our aim is to prove that carbon dioxide and heat is released during anaerobic respiration. Operators. For this activity, you need thermoflask, spilt cork, thermometer, wash bottle, glass tubes, liquid paraffin wax, glucose solution, yeast cells, bicarbonate solution. These are the operators for activity. Procedure. First of all, you have to take glucose solution in a flask, and after remove dissolved oxygen from glucose solution by boiling it in thermoflask for a minute and then cool it without shaking. So whenever we are heat the solution, in the heating time, oxygen removing from the glucose solution. With using of heating, we will remove oxygen from the glucose solution. And after that, you have to add some yeast cell to the glucose solution and fix 2-hole rubber cork stopper to the flask. After removing oxygen, you have to add yeast cells, and after that you have to attach the 2-hole rubber cork to the flask. The supply of oxygen from the outside atmosphere to be cut off by pouring 1 centimeter layer of liquid paraffin wax. After that, insert one end of the thermometer into the thermos flask. See the end of the thermometer kept inside of the solution. So we have to use one thermometer. We want to insert the thermometer into the flask with using of one hole in rubber cork. After that, insert the end of the thermometer into the thermo flask. See the end of the thermometer kept into the inside of the solution. Arrange for any gas produced by the yeast to escape through the wash bottle containing bicarbonate solution or lime water. It is shown in the picture. After that, the blue diazine green solution turns into pink when oxygen is short supply around it. Whenever we are added diazine solution into the glucose solution, it is turns into the pink color. Whenever it is turns into the pink color, it is indicating there is no oxygen in the glucose solution. After that, warm the operators to about 37 degrees Fahrenheit and it is important for speed of the activity of yeast cells. And after that, keep the operator undisturbed for 1 or 2 days. After 2 days, you have to observe. In this activity, we are observed that increasing in temperature noted on the yeast cells respire and releasing energy. Increasing in temperature noted on yeast cells respire and releasing energy. Alcohol smell given or from the flask. So we will get alcohol smell from the flask. And also lime water turns into the milky white. It is present in the wash bottle. In this activity, we are conclude that our result is these observations indicate that the yeast cells respire anaerobically, converting glucose solution into carbon dioxide and ethyl alcohol, and also releasing heat energy. This heat energy we are observed in raising temperature in thermometer. This is the activity for testing of anaerobic respiration or fermentation in our school laboratory. In this activity, you have to take some precautions. Ensure that oxygen does not enter into the glucose solution. And also, thermometer must attached with the glucose solution. These 2 precautions you have to take in this activity. Our next topic is differences between the respiration and combustion. First of all, I want to explain about respiration. Respiration occurs in the living cells. It occurs at the body temperature. Energy is released in several stages. Several intermediates are formed when respiration occurs in our body. For example, carbon dioxide, water, heat. Enzymes are required for respiration. It occurs in the presence of water. It is controlled process. Respiration is a controlled process. Energy is stored in ATP in the body. So when we are seeing these points with combustion, how it is different from one to another. Respiration occurs in the living bodies, but combustion occurs in the non-living cells. Respiration occurs with the help of our body temperature, but heat is applied for combustion process. Energy is released in several stages in respiration, but in the combustion, energy is released at once as a heat in the form of heat. There are production of several intermediates in respiration. Example, carbon dioxide, heat, water. There is no production of intermediates in combustion process. Respiration occurs with the help of enzyme. There is no use of enzyme in the combustion process. Respiration occurs in the presence of water because of in our cells we have cytoplasm. Cytoplasm is in liquid state. So in that medium also respiration occurs. But combustion process is not occurs in the presence of water. Respiration is a controlled process. When we are not providing oxygen to the cells, respiration will stop. But it is uncontrolled process when it is started. Respiration is energy production process. That energy is stored in the body in the form of ATP. Energy is not stored in combustion. So these are the differences between the respiration and combustion. And also we are seeing some similarities between the respiration and combustion. In these 2 processes, glucose is utilizing, and also oxygen is utilized in respiration and also combustion. And final products are production of energy and carbon dioxide. These are the similarities we are seeing in the combustion process and also respiration process. Second activity in second chapter, observing changes during combustion of sugar or glucose. So whenever we are heating the sugar, what kind of changes we are seeing? For that, we want to do one activity. First of all, you have to take one test tube and fill it with some glucose or sugar or flour. And after, you have to take one whole rubber cork. In that hole, you have to fix a delivery tube. And after that, you have to arrange this test tube to the stand. And after, you have to arrange the delivery tube into the test tube. It is contain lime water. After that, with using of burning splinters or burner, you have to heat the glucose present in the test tube. Whenever you are started burning or heating glucose in the test tube, it is producing some fume like gas. It is enter into the lime water with the help of delivery tube. And also we are seeing small droplets are present in the test tube walls. So in this activity we are observing mainly two. Lime water turns into the milky white and some droplets are present in the test tube walls, first test tube walls. From this activity we are conclude that when we are burning glucose, it is producing water and carbon dioxide. Water, it is present in the test tube walls and carbon dioxide enter into the lime water, it turns into the milky white. These are the observation in this activity. Whenever we are burning or whenever we are heating glucose solution, it is producing carbon dioxide and water.